Long before Earth became the blue world seen from space, it wore another face. More than 2.5 billion years ago, there were no forests leaning into the wind, no grasslands, no wings crossing the sky, and no animals moving through the sea. The continents were darker, smaller, and more barren. Volcanoes opened across their surfaces, releasing gases into an atmosphere that no human being could survive. Above them, the young sun shone with less strength than it does today, yet oceans still covered much of the planet. From a distance, this world might have looked calm. Up close, it would have seemed less like home and more like an unfamiliar planet wearing Earth's name. The sky itself may not have carried the clear blue color now taken for granted. Blue daylight depends partly on the way sunlight moves through the modern atmosphere, but this ancient air held a very different mixture of gases. Nitrogen was abundant. Carbon dioxide, water vapor, methane, and volcanic gases helped shape the climate. Free oxygen was almost absent. A visitor removing a helmet would not have felt a moment of discomfort before recovering. There would have been no recovery. The lungs would search for a gas that simply was not there. Yet the absence of oxygen did not mean the absence of life. That is the first illusion the ancient Earth asks us to abandon. Modern life has trained the human mind to connect breathing with living, as though oxygen were the beginning of every biological story. But life had already existed for an immense span of time before the atmosphere became breathable. It occupied shorelines, shallow seas, deep water, warm springs, mineral surfaces, and cracks beneath the ocean floor. It did not wait for the modern world. It built another one. Across iron-rich seas, microscopic communities gathered in thin films and layered mats. They used chemical reactions that would seem strange to most organisms alive today. Some drew energy from hydrogen, others worked with sulfur, iron, carbon dioxide, or methane. They lived without lungs, without blood, and without the steady supply of oxygen that would later power large bodies. Their world was not incomplete. It was complete according to different rules. Those rules reached into the rocks themselves. The surface of the planet contained enormous amounts of material ready to react with oxygen. Iron dissolved through seawater. Fresh volcanic minerals lay exposed. Gases rising from Earth's interior carried chemical energy that could quickly consume any oxygen that appeared. The planet was not merely lacking oxygen, it was prepared to destroy it. Imagine standing on a black volcanic shore beneath that strange sky. Waves move across a dark ocean, carrying dissolved metals invisible to the eye. The land has no roots to hold it together. Rain falls on naked stone and runs back toward the sea. There is no bird call, no insect movement, and no leaf turning toward the light. The silence is not the silence of death. Beneath the surface, countless cells are feeding, dividing, competing, and exchanging chemical products. The planet is alive, but almost all of its life is hidden. This hidden biosphere changes the meaning of the word alien. Human beings often look toward distant stars and imagine worlds with unfamiliar atmospheres and strange forms of life. But Earth itself passed through such a condition. For much of its early history, it would not have recognized the needs of the creatures that now study it. A modern person would be an impossible organism there, dependent on a future atmosphere that had not yet been invented. The most remarkable part is that this world endured. It was not a brief opening scene before the important history began. For hundreds of millions of years, life adapted to oxygen-poor conditions and spread through them. Entire lineages rose, changed, and disappeared before any animal took a breath. Their chemistry helped control the oceans and atmosphere. Methane produced by microbes may have warmed the planet beneath the faint young sun. Other organisms altered sulfur and iron, leaving traces that would survive in stone long after the cells themselves vanished. Geologists searching those stones do not find a simple diary. They find fragments, minerals that formed under certain conditions, isotopes arranged in unusual patterns, sediments laid down beneath ancient water, and chemical signals that appear, shift, then disappear. From these fragments, a picture emerges. Before the great rise of oxygen, the atmosphere contained only tiny amounts of the gas. Local traces may have existed, especially near organisms producing it, but the wider sky remained almost entirely without it. This distinction matters. Oxygen did not suddenly appear from nothing on one dramatic day. Long before it accumulated in the air, Small quantities were probably being made and immediately removed. The ancient planet behaved like a vast room with countless open drains. Every new drop vanished before a pool could form. The mystery is not only who produced the oxygen, the deeper mystery is what eventually closed enough of those drains for the gas to remain. For the organisms already living there, that question would have had no meaning. They did not know the atmosphere was approaching a turning point. They did not see themselves as early life waiting to be replaced. They were the rulers of their age, perfectly suited to seas and sediments that had supported them for longer than complex life has existed. And somewhere within that quiet microbial world, a new way of using sunlight was taking shape. It would release a substance that the planet had spent ages removing. At first, the change would be almost invisible, a few molecules made in shallow water and lost to iron, rock, and gas. But the process would continue. Day after day, generation after generation, life would begin producing the very thing that could end the world it knew. The greatest danger was not hidden in a volcano or falling from space. It was rising quietly from cells beneath the ancient sun. The shore gives way to shallow water, and the ancient world becomes crowded, not with fish or drifting plants, but with layers of microscopic life. Along mineral surfaces, cells gather into films. In warm pools and tidal flats, they build mats with one community resting above another. The upper layers meet sunlight. Lower layers touch sulfur, iron, and gases rising from sediment. Each narrow zone holds a different form of opportunity. To modern eyes, these organisms seem simple because they are small. That judgment is misleading. Their cells contain systems refined across immense periods of evolution. They sense changes in light and chemistry. They compete for nutrients. They release substances other organisms can use. Some live beside one another because the waste of one becomes the food of another. Others defend territory through chemical means. There are no teeth or claws, yet the struggle for survival is already ancient. Much of this life is anaerobic. The word simply means that it lives without free oxygen. Some anaerobic organisms can endure a little oxygen, but others are harmed or killed by it. Their enzymes were shaped in a world where oxygen was rare. When exposed to the gas, delicate chemical machinery could be disrupted. Dangerous reactive forms of oxygen could damage membranes, proteins, and the material carrying genetic instructions. What later animals would draw into their lungs was, for many early organisms, a form of poison. This is difficult to imagine, because oxygen now surrounds nearly every familiar ecosystem. It fills the atmosphere, mixes into rivers, reaches the upper ocean, and enters soil through roots and burrows. Even creatures that never breathe air often depend on oxygen dissolved in water. The modern biosphere has been built around the gas so completely that its absence feels like a missing foundation. But on the early Earth, oxygen was the intruder. In deeper mud, methane-producing archaea converted simple carbon compounds into methane. Their activity may have helped keep the planet warm. Elsewhere, microbes use sulfate or sulfur compounds in reactions that moved elements between water and sediment. Some organisms interacted with dissolved iron. Others survived far below the surface, where water moved slowly through cracks in warm rock. The biosphere extended through many environments, but nearly all of it remained invisible from a distant view. No single species ruled this planet in the manner humans now imagine themselves ruling the modern world. Instead, control was distributed across countless microbial populations. Together, they shaped the atmosphere, altered minerals, and controlled the movement of carbon. Their influence was enormous, precisely because their numbers were enormous, and their activity never stopped. The ancient ocean was not chemically uniform. Near volcanic regions, new substances entered the water. Around hydrothermal vents, hot fluids carried metals and gases from the crust. In shallow seas, sunlight powered photosynthetic reactions that did not necessarily release oxygen. Beneath microbial mats, the chemistry could change within the width of a finger. One layer might use light, another might consume sulfur, a deeper layer might produce methane. The mat was a small world arranged by chemistry. These communities left few recognizable bodies behind. Microbes do not usually form bones, shells, or large structures that survive easily for billions of years. Some mats created layered formations known as stromatolites. Other organisms influenced minerals or left chemical signatures in sediment. But much of their diversity disappeared without a clear fossil record. This absence creates a problem when the story reaches the coming catastrophe. Later mass extinctions can be measured through the sudden disappearance of many fossil species. The end of the dinosaurs is written in bones, pollen, shocked minerals, and a thin global layer linked to an impact. The oxygen crisis left no such complete list of victims. Its casualties were microscopic, their ecosystems ancient, and much of the relevant seafloor has been destroyed or transformed by geology. So when this event is called Earth's first mass extinction, the phrase must be understood carefully. It describes a likely global loss of habitat suitable for oxygen-sensitive life, together with the disappearance, retreat, or replacement of many microbial lineages. It does not mean scientists can count exactly how many species vanished. The evidence is indirect, but the biological logic is powerful. When a planet's atmosphere and surface ocean gain a substance toxic to much of the existing biosphere, ecological destruction is not a dramatic guess. It is a probable consequence. Still, extinction was not the only possible response. Some organisms moved downward into sediment. Some remained in deep water where oxygen could not easily reach. Others occupied protected pockets beneath rock or within dense microbial communities. The old world was pushed back, not erased. Its descendants continue today in wetlands, digestive systems, deep mud, hot springs, and the crust beneath the oceans. That survival carries a quiet message. Ancient life was not weak. It endured the greatest chemical transformation the planet had yet experienced by finding places the new atmosphere could not control. Before that retreat began, however, the anaerobic world remained vast. Methane moved upward from the sea. Iron circulated through oxygen-poor water. Sunlight reached microbial communities that had used it for ages without changing the sky. The planet appeared stable because every process had become part of a long balance. Yet stability can hide the machinery of its own ending. Within this invisible empire, one lineage was learning to take electrons from a substance found almost everywhere. It did not need rare sulfur compounds or limited deposits of dissolved iron. It could use water. That achievement would give its descendants access to a nearly endless source of chemical power. The leftover product seemed small, pairs of oxygen atoms released into the surroundings. Most vanished quickly. Some may have harmed nearby cells. None appeared capable of overturning the planet. But the new organisms did not need to understand what they were doing. They only needed sunlight, water, and time. The ancient Earth had all three in abundance, and time would prove more dangerous than intention. Sunlight had touched the ancient oceans for billions of mornings, but life did not discover only one way to use it. Long before oxygen became common, microbes had already learned to capture light and turn it into chemical energy. These early forms of photosynthesis worked without releasing the gas that now fills the atmosphere. They borrowed electrons from substances such as hydrogen sulfide, hydrogen, or dissolved iron. The method was effective, but the needed materials were not equally available everywhere. Water was different. It covered most of the planet. It filled seas, fell as rain, moved through rock, and surrounded every cell living near the surface. If an organism could draw useful electrons from water, it would gain access to a source so widespread that earlier photosynthetic systems could scarcely compete with it. There was one problem. Water is chemically stubborn. Its molecules hold their parts together with remarkable strength. Breaking them in a controlled way requires energy and a carefully arranged system of reactions. A cell cannot simply tear water apart without risking damage or wasting the energy it hoped to capture. The solution that evolved was among the most complex pieces of molecular machinery in the early biosphere. Ancestral oxygen-producing microbes developed linked systems that absorbed light in stages. Pigments caught the energy of photons. Proteins guided electrons through membranes. At the heart of the process, a small cluster containing metal atoms helped remove electrons from water. The cell used those electrons to build energy-rich compounds and organic matter. Oxygen emerged from the reaction as leftover material. That final detail would eventually change the planet, but it was not the purpose of the process. The microbes were not trying to prepare Earth for animals. They were not moving toward forests, lungs, or human thought. Evolution has no distant plan. Each generation succeeds only by surviving and reproducing under the conditions immediately around it. Water-splitting photosynthesis gave its users a powerful advantage. Sunlight was abundant in shallow seas, and water did not run out in the way local supplies of sulfur or iron could. Cyanobacterial ancestors could spread across illuminated environments, forming films, floating communities, and layered mats. Their waste entered the water one molecule at a time. The familiar name for this process is oxygenic photosynthesis. It now occurs in plants, algae, and cyanobacteria. In plants, the work takes place inside chloroplasts, structures descended from ancient cyanobacteria that entered into partnership with other cells much later. But the first oxygen producers were microscopic. The world-changing reaction began without leaves, roots, flowers, or green continents. Exactly when it began remains uncertain. Some geological signals suggest small amounts of oxygen existed long before the atmosphere permanently changed. Molecular studies of photosynthetic proteins also point toward deep origins. Yet ancient rocks have been heated, buried, broken, and altered. Chemical traces can have more than one explanation. Scientists therefore debate whether oxygenic photosynthesis arose hundreds of millions of years before the Great Oxidation Event, or closer to the atmospheric transition itself. The uncertainty makes the story more interesting, not less. If oxygen production began early, then the planet resisted it for an extraordinary length of time. The organisms may have been releasing oxygen through countless generations, while oceans, rocks, and volcanic gases consumed every trace. A revolution could have been happening in daylight, without leaving the sky visibly changed. At first, oxygen would have been a local danger. A cyanobacterial mat producing the gas might create a thin, oxygen-rich layer around itself. Nearby anaerobic organisms would face a choice they could not consciously understand: retreat, tolerate the exposure, exploit the new chemistry, or die. The first oxygen crisis may have occurred not across the entire planet, but within microscopic distances inside living mats. One layer released oxygen, another consumed it. Iron in the water reacted with it. Organic matter absorbed its chemical power during decay. The new gas entered an environment crowded with substances ready to remove it. Its global concentration stayed low, but its biological influence could begin long before atmospheric instruments would have detected it. This changes the usual picture of the oxygen revolution. It was not simply a moment when cyanobacteria appeared and the air transformed. Biological invention and planetary transformation were separated by time. The machinery came first. The atmosphere followed only after Earth's chemical defenses began to weaken or fill. The waste product itself was also more dangerous than its ordinary name suggests. Oxygen reacts eagerly. In controlled systems, that eagerness makes it useful. In unprotected cells, it can create reactive molecules that attack essential structures. The organisms releasing oxygen may have needed defenses against their own product, especially when sunlight was strongest. Their survival required balance. They had to make use of light, handle powerful reactions, repair damage, and compete with organisms using older forms of photosynthesis. Nothing about their success was guaranteed. Yet once water splitting had evolved, the possibility of a different planet existed. For a long time, possibility remained trapped close to the organisms that created it. Oxygen appeared during the day and vanished into the surrounding world. The atmosphere above remained almost unchanged. Methane-producing microbes continued to influence the climate. Iron-rich seas continued to absorb the new gas. The anaerobic empire still seemed secure. But the invention could not be uninvented. Each cell division copied the machinery. Each new population carried it into another sunlit place. Each morning began the reaction again. The greatest transformation in Earth's atmospheric history started not with an explosion, but with a quiet improvement in the way a microbe fed itself. What later life would call breathable air began as unwanted waste, released by organisms too small to see and too successful to stop. And still the ancient sky gave no sign of surrender. The gas entered water, met iron, touched volcanic chemicals, and disappeared. To any observer, nothing important had happened. Yet beneath the surface, life had discovered a reaction that could be repeated wherever sunlight reached the sea. If the ancient atmosphere remained almost without oxygen, the first question seems obvious. Where did all the gas go? The answer begins by changing the scale. Instead of looking at the entire planet, the The story moves down to a patch of shallow water no wider than a small room. A microbial mat lies across the bottom. Its surface receives sunlight. During the day, oxygen-producing cells begin their work. Light enters pigments, electrons move through membranes, water is split, and oxygen leaks into the water around them. For a few hours, the concentration rises faster than nearby reactions can remove it. A thin oxygenated zone forms above the mat. This is not yet an oxygenated ocean. It is an oasis. A short distance away, the water may still contain almost no free oxygen. Beneath the mat's surface, other microbes continue living anaerobically. They consume organic matter, use sulfur compounds, or produce methane. Across a depth of only millimeters, the chemistry shifts from a preview of the modern world to conditions inherited from the ancient one. At sunset, the balance changes. Photosynthesis slows and stops. Cells continue using energy. Organic matter continues to decay. Iron and other reduced substances continue reacting. The oxygen made during daylight is consumed. By night, the oasis contracts or disappears. Then morning returns, and the cycle begins again. These daily pulses offer a way to understand how oxygen-producing life could exist long before the atmosphere changed. The gas did not need to fill the sky to influence evolution. It only needed to appear often enough in local habitats. Organisms living nearby would experience repeated exposure. Some would be damaged, some would move into deeper layers, some would evolve defenses. A few might discover ways to use oxygen as part of their own metabolism. The boundary between oxygen-rich and oxygen-poor water became an ecological frontier. Along that boundary, chemical energy was available because substances from two different worlds met. Reduced compounds rose from sediment. Oxygen moved downward from the sunlit surface. Microbes positioned between them could take advantage of the reaction. This kind of boundary still exists today. In lake mud, wetlands, ocean sediments, and microbial mats, oxygen may disappear within a short distance. Different organisms occupy different layers according to what they can tolerate and consume. Modern environments provide a small window into processes that once dominated the planet. The ancient version, however, unfolded within iron-rich seas. Dissolved iron could enter the oxygen oasis and react with the gas. Oxidized iron particles then settled downward, carrying the evidence into sediment. The same iron also created trouble for oxygen-producing cells, especially under strong light, because chemical reactions could generate damaging molecules. Cyanobacteria had to survive not only the old biosphere, but the reactive ocean itself. Some researchers have recreated parts of these conditions in laboratories. They expose modern cyanobacteria to iron-rich water, changing light and daily cycles. The results suggest that oxygen production and iron oxidation can occur together in complex ways. The microbes may create localized oxygen, while most of the surrounding water remains anoxic. The system breathes with the sun, expanding in daylight and collapsing in darkness. Now imagine this pattern repeated across thousands of coastlines and shallow seas. Each oasis is small. None transforms the atmosphere alone, but together they produce oxygen every day. Some oxygen reacts immediately, some is trapped in minerals. Some supports new metabolisms. A tiny fraction may escape into the air before being consumed by gases or weathered rock. Already, at the scale of a microbial mat, the future has arrived in daylight. This unevenness helps explain why geological evidence before the Great Oxidation Event can appear confusing. One rock formation may preserve signs of local oxygen. Another, formed at nearly the same time, may record strongly oxygen-poor conditions. Both can be true. Earth was not moving as a single uniform body. Its surface contained chemical islands, temporary zones, and shifting boundaries. The idea of oxygen oases also changes the story of survival. Anaerobic life did not wake one morning beneath a completely transformed sky. The threat approached in patches. Shallow habitats became dangerous before deep ones. Daylight exposure came before permanent exposure. Organisms experienced the revolution at different times depending on where they lived. For oxygen-producing microbes, the oases were both shelter and prison. Inside them, they could use their powerful metabolism. Outside them, the wider ocean erased the gas they made. Their success increased local oxygen, but each expansion brought them into contact with more iron, more reduced chemicals, and more biological competitors. The planet seemed to possess endless defenses. Oceans held dissolved material ready to react. Volcanoes supplied gases that consumed oxygen. Newly exposed rock drew the gas from air and water. Organic matter captured sunlight, then used oxygen again when it decayed. Every route led back toward removal, and yet the oxygen oases mattered because they created memory. Minerals changed, genes spread, new ecological relationships formed. Organisms adapted to a gas the atmosphere still barely contained. The world was being prepared in pieces. The strange part is how long this preparation may have lasted. Oxygen-producing photosynthesis could have preceded permanent atmospheric oxygen by hundreds of millions of years. If so, countless sunrises created the same brief future, and countless nights closed over it. But chemical defenses are not infinite. Iron can be oxidized, reduced surfaces can change, volcanic gases can alter as Earth's interior evolves. Organic carbon can be buried beyond the reach of decay. Slowly, the balance between production and removal can shift. The microbial mats did not know they were filling planetary sinks. They simply returned to the light each morning. Yet every oxygen molecule that became locked into stone rather than consumed in water removed one small part of Earth's resistance. The oases were temporary, but the changes beneath them were not. Somewhere beyond their thin boundaries, the ancient planet was approaching a point when night would no longer erase everything daylight had made. Beyond the thin oxygen oases, the larger planet continued to behave like a machine, built to remove the gas. Every direction offered another reaction. The oceans held dissolved iron. The crust exposed fresh minerals. Volcanoes released hydrogen, sulfur compounds, carbon monoxide, and other reduced gases. Dead cells sank into water and sediment, where their organic matter could be broken down. Oxygen entering this system was not entering empty space. It was entering a crowded chemical market in which almost everything wanted to react with it. The iron-rich ocean provides the clearest image. In many ancient seas, iron existed in a dissolved form that remains stable only when oxygen is scarce. When oxygen reached it, the iron changed. It became less soluble, formed particles, and settled toward the seafloor. Over time, iron-rich layers accumulated with silica and other sediments. Pressure, heat, and geological change transformed them into the banded iron formations, mined and studied today. These rocks often appear as repeated dark and red bands, as though the planet kept a striped record of its own chemistry. Their beauty can make the story seem simple. Cyanobacteria released oxygen, the oxygen rusted the ocean, and the rust fell to the bottom. Some of that picture is useful, but the complete process was more complicated. Iron could be oxidized in several ways. Oxygen produced by microbes may have reacted directly with dissolved iron. Light-driven reactions could also alter iron without free oxygen. Other microbes may have used iron in their metabolism. Ocean circulation, silica content, temperature, and the supply of iron from hydrothermal systems shaped where deposits formed. Banded iron formations are evidence of a vast iron cycle, not a single reaction repeated without variation. Still, they reveal the scale of the oxygen sink. Before oxygen could remain in the atmosphere, enormous quantities of reduced iron and other materials had to be altered. The gas made by living cells disappeared into minerals, leaving stone where breathable air might otherwise have formed. A simple way to imagine this is as a planetary account. Photosynthesis adds oxygen. Every oxidation reaction subtracts it. Iron in the sea is a major expense. Volcanic gases are another. Weathering of fresh rock draws down more. The decay of organic matter can return carbon to the environment while consuming oxygen that photosynthesis released. As long as spending equals or exceeds income, the atmospheric balance remains near zero. The accounting metaphor hides one important detail. The expenses change with time. An iron atom oxidized and buried cannot consume oxygen forever. A mineral surface that has already reacted becomes less powerful than a fresh one. Volcanic gases shift as Earth's mantle cools and the crust evolves. Continents grow, erosion changes, and sediments bury organic material beyond easy contact with oxygen. The planet's ability to resist oxygen was enormous, but it was not endless. For hundreds of millions of years, oxygen-producing organisms may have paid into this chemical system without seeing the atmosphere change. Their work oxidized coastlines, altered ocean minerals, and created new ecological boundaries. The oxygen was not wasted in a geological sense. It was changing the reservoirs that controlled the future atmosphere. This hidden transformation explains why the eventual rise of oxygen could appear sudden. A sink can absorb material quietly until it begins to fill. A dry sponge does not create a puddle when the first drops fall, but once saturated, each new drop remains on the surface. Early Earth was not a single sponge, and its sinks were never completely full, yet the principle helps reveal the threshold. Fresh oxygen also affected sulfur. It changed how sulfur compounds moved between air, water, and sediment. It could release certain metals from rocks, while causing others to precipitate. Nutrients became more available in some places and less available in others. Toxic elements could enter water as weathering intensified. The change was not merely a rising number in the atmosphere, it was a reorganization of the entire surface chemistry of Earth. The most active sinks existed where worlds met. Oxygen from shallow water encountered iron from deeper water. Rain carried small amounts of oxygen toward bare continental rock. Volcanic gases rose into an atmosphere beginning to contain a new reactant. At each boundary, oxygen vanished while leaving altered matter behind. To the microbes producing it, none of this planetary bookkeeping was visible. They experienced only local conditions: light, nutrients, competitors, heat, and the chemistry immediately surrounding their cells. Yet their combined activity forced oceans and rocks to absorb more oxygen than any earlier period of Earth history. The stone record preserves this pressure. Vast iron deposits show that the oceans did not remain chemically untouched while the atmosphere stayed oxygen poor. Something was driving oxidation on a scale large enough to reshape sediments across continents. Then, across the long approach to the Great Oxidation Event, the nature of the sinks began to change. Some reduced reservoirs became less able to remove oxygen. Organic carbon was buried in ways that left oxygen behind. The gases coming from Earth's interior may have become less strongly reducing. Biological communities altered the supply of methane and nutrients. No single switch explains everything. Instead, several slow changes approached one another. Oxygen production continued. Chemical resistance weakened. The balance moved toward a line no organism could see. Among the possible changes, one involves an element rarely associated with the atmosphere at all. Nickel, present only in small amounts, was essential to organisms that made methane. If its supply declined, the consequences could reach from enzymes inside cells to the temperature of the entire planet. The next clue in the oxygen mystery is therefore not found in the gas itself. It is found in the weakening of the microbes that helped keep oxygen away. The oceans had been hiding the revolution inside rust, sediment, and altered crust. Once enough of that resistance changed, the same daily oxygen production that once vanished without consequence could begin leaving a permanent mark above the waves at last. The ancient atmosphere was not controlled by oxygen alone. Another gas, produced by organisms living without oxygen, may have helped keep the planet warm and the new gas suppressed. That gas was methane. Methane-producing archaea lived in sediments, shallow waters, and other environments where oxygen was absent. They used simple compounds and released methane as a product of their metabolism. Once the gas entered the atmosphere, it trapped heat far more effectively than carbon dioxide molecule for molecule. This may have helped solve one of the oldest climate puzzles: how liquid oceans survived beneath a young sun that shone more faintly than the sun does today. A methane-rich atmosphere also made oxygen accumulation difficult. When oxygen entered the air, it reacted through a chain of processes that destroyed methane. The two gases could not remain together in large amounts without continuous replacement. As long as methanogens produced methane rapidly and Earth's surface contained many reduced materials, oxygen remained under pressure from every side. The survival of methanogens depended on more than carbon and water. Deep inside their cells, specialized enzymes carried out the final steps of methane production. Some of those enzymes required nickel. Nickel is not abundant in seawater, yet a small supply can govern the activity of organisms spread across an ocean. The element entered ancient seas through volcanic material, hydrothermal systems, erosion, and chemical weathering. The mantle produced rocks richer in nickel than many later volcanic rocks. As the planet cooled, the composition of volcanic material changed. Less nickel may have reached the oceans. In 2009, researchers proposed that this decline created a nickel famine for methane-producing microbes. With less nickel available, methanogens would make less methane. The atmospheric shield would weaken, reducing one of the major forces opposing oxygen. The idea is powerful because it connects four scales that rarely seem related. A change deep inside the cooling Earth alters volcanic rocks. Those rocks change the trace metals entering the sea. The trace metal affects an enzyme inside a microscopic cell. The cell changes the atmosphere and climate. It is exactly the kind of hidden connection that makes ancient Earth difficult to reconstruct. No single observer could have watched the entire chain. Yet the nickel famine is not a complete answer. Later studies suggest that weathering of sulfide minerals on land may have continued supplying nickel to some oceans. Different basins received different amounts. Methanogens did not disappear, and methane production did not simply stop. The decline may have been regional, gradual, or only one part of a larger change. Other factors were moving at the same time. The chemistry of volcanic gases may have become more oxidized. Continents were growing and shifting. Weathering carried nutrients into shallow seas. Organic carbon was buried in sediment, allowing some oxygen to remain. Cyanobacteria may have expanded as ecological conditions improved. Iron and sulfur sinks continued evolving. The Great Oxidation Event was probably not caused by one dramatic invention followed by one dramatic trigger. It emerged from a network of changes that gradually made the old atmospheric balance harder to maintain. Still, methane holds a special place in the story. It was both biological and climatic. Methanogens helped support the warm world in which early oxygen producers evolved. Their gas also reacted with oxygen, delaying its rise. The older anaerobic biosphere was not merely waiting to be poisoned. It was actively shaping the planet in ways that protected its own conditions. Then its protection began to weaken. Imagine a shallow sea, where nickel becomes harder to obtain. Methanogenic communities grow more slowly. Less methane rises through the water. At the same time, cyanobacterial mats continue producing oxygen in daylight. Iron still removes much of it, but not every sink is as powerful as before. More organic material is buried. A slightly larger fraction of oxygen survives each cycle. Nothing about this moment would appear dramatic. There is no visible line across the ocean. No alarm sounds among the microbes. The atmosphere changes through tiny imbalances repeated over immense time. If oxygen production exceeds removal by even a small amount, the difference accumulates. A fraction becomes a trace becomes a persistent presence. Persistent oxygen changes weathering, sulfur chemistry, and the survival of methane. Each new change can reinforce the next. This is where the planet begins to behave less like a stable background and more like a system approaching a threshold. For ages, feedbacks held oxygen down. Now some feedbacks begin turning in the opposite direction. Oxygen destroys methane. Lower methane may cool the planet and alter ecosystems. Oxygen changes minerals, affecting nutrients and metal availability. The biological world responds, changing gas production again. The methane shield was never a literal layer surrounding Earth. It was a balance maintained by living cells, chemical reactions, and continuous replacement. Once that balance weakened, the atmosphere could move into a state the anaerobic world had never experienced. The irony is sharp. Methane-producing organisms had helped warm and protect early life. Oxygen-producing organisms used the same sunlight and oceans to create a competing atmospheric future. Neither group understood the struggle. Their metabolisms simply altered the conditions inherited by every generation after them. Scientists continue debating which factor mattered most. Nickel may have played a supporting role rather than the leading one. Tectonics, nutrient cycles, carbon burial, and changes in volcanic gases may have been equally important or more important. The correct picture may differ across regions and through time, but the central truth remains. Oxygen rose not only because its producers became successful, but because the world consuming their product changed. The gas had been pressing against a planetary barrier for millions of years. Methane was part of that barrier. When it weakened, the pressure did not need to increase much before something gave way. The coming threshold would leave no footprint, bone, or ruin. It would leave a disappearance in sulfur, a change in iron, and a new chemical logic written into rocks around the world. The atmosphere of the distant past cannot be placed inside a bottle and carried into a laboratory. No bubble of air has survived unchanged for more than 2 billion years. Yet the ancient sky left a record, not in the form of trapped breath, but in the way sunlight altered sulfur. Sulfur moves easily between volcanoes, atmosphere, ocean, organisms, and sediment. It can exist in several chemical forms, and its atoms occur in different masses, called isotopes. Most natural processes separate those isotopes according to their weight. Lighter atoms react slightly faster than heavier ones, producing patterns scientists can predict. Before oxygen became persistent, something unusual happened high in the atmosphere. Ultraviolet light struck sulfur-bearing gases released by volcanoes. Under oxygen-poor conditions, these reactions sorted sulfur isotopes in ways that did not follow the ordinary rules of mass. The result is known as mass-independent fractionation, but the name matters less than the clue it preserves. The ancient atmosphere was creating a sulfur pattern that modern oxygen-rich air does not. Rain and particles carried the altered sulfur into the ocean. Microbes used it, minerals captured it, sediments buried it. Billions of years later, geologists could measure the unusual pattern in tiny samples of rock. Then, near the time of the Great Oxidation Event, the signal largely vanished. The disappearance is one of the most important pieces of evidence in the entire story. Once oxygen rose beyond a low threshold, atmospheric chemistry changed. An ozone layer began to form from oxygen higher in the atmosphere, reducing the ultraviolet light available for the old sulfur reactions. Sulfur compounds followed different pathways and were mixed more thoroughly before reaching sediments. The strange isotope signature could no longer be preserved on a wide scale. This creates a rare kind of proof. Scientists identify the arrival of a new atmospheric state through the loss of a chemical pattern. The missing signal becomes the message. The transition was not perfectly clean. In some rock sequences, the unusual sulfur signature weakens, returns, and weakens again. Different sedimentary basins appear to record the change at slightly different times. Some of these differences may reflect real regional conditions. Others may come from uncertainty in dating, later alteration of rocks, or gaps in deposition. The ancient atmosphere may have crossed the oxygen threshold several times before remaining on the new side. A pulse of oxygen could reduce the sulfur signal. A later increase in volcanic gases or oxygen-consuming reactions might restore oxygen-poor conditions. The planet may have moved forward and backward, testing the boundary before the change became permanent. That possibility challenges the familiar image of a single great oxidation event. The word event suggests one moment, like an eruption or impact. In reality, the transformation may have involved a long approach, several unstable steps, and a final shift, after which the old atmospheric state could no longer return completely. Geologists reconstruct this sequence by comparing distant rock formations. In South Africa, ancient sediments preserve sulfur isotope changes alongside evidence from carbon and iron. In Western Australia, other formations reveal oxygen conditions before and during the transition. North American rocks add another timeline. Each location is incomplete, but together they form a planetary puzzle. The puzzle requires caution. A chemical signal can be influenced by local water chemistry, microbial activity, burial, heating, and later fluids moving through rock. Scientists test multiple explanations, compare independent indicators, and revise dates when new measurements appear. The picture grows sharper, but it never becomes a recording watched in real time. This uncertainty does not weaken the central conclusion. Before the Great Oxidation Event, the sulfur pattern could form and survive because atmospheric oxygen was extremely low. After the transition, that pattern mostly disappeared because the atmosphere had crossed into a different chemical regime. The sky had changed enough to rewrite the behavior of sunlight. Imagine the transformation from the perspective of the sulfur atoms themselves. For ages, volcanic gases rose into an atmosphere open to intense ultraviolet reactions. Their sulfur was divided into unusual isotope groups, then scattered into seas and stone. Later, the same gases entered air, containing more oxygen and ozone. The old pathway closed. A signature repeated across millions of years stopped appearing. No mountain split in two when this happened. No creature watched the sky change color in a single lifetime. The threshold became visible only in retrospect, through instruments capable of measuring differences smaller than anything ancient life could sense. Yet for organisms living near the surface, the consequences were immediate on their own scale. More oxygen in air meant more oxygen entering water. Oxidation reached minerals that had remained reduced for ages. Methane became less stable. Anaerobic habitats near coastlines and shallow seas contracted. A chemical change detected by sulfur isotopes represented a biological change experienced by entire ecosystems. The sulfur record also shows why oxygen's rise cannot be understood only by finding the oldest evidence of photosynthesis. Oxygen-producing microbes may have existed long before the atmospheric signal disappeared. The crucial moment came when production, burial, weathering, volcanic chemistry, and biological consumption combined to push the atmosphere past a threshold. What had been local became persistent. What had appeared only in daylight oases began surviving through the night and spreading beyond the water that produced it. The signal's disappearance therefore marks more than the arrival of oxygen. It marks the failure of the old planet to erase oxygen completely. The remaining question is how quickly that failure unfolded. Geological time can make every change seem slow, but improved dating suggests that one decisive rise may have occurred within a surprisingly narrow interval. For Earth, the revolution may have been sudden. For life trapped inside it, even 1,000,000 years could be long enough for adaptation, extinction, and entire worlds to vanish without a witness. The rocks do not reveal a single sunrise when the atmosphere changed. They reveal something stranger, a boundary crossed repeatedly, then held, until chemistry itself remembered oxygen permanently. Human beings are poor at feeling geological time. A century can seem distant. 1,000 years becomes history. 1,000,000 years dissolves into abstraction. Yet when Earth is more than 4 billion years old, 1,000,000 years can represent a sharp turn. The Great Oxidation Event is often described as though the atmosphere slowly brightened across an endless age. In one sense, that is true. Oxygen-producing life may have existed long before the gas became persistent, and the full transformation unfolded through several stages. But high-resolution dating of ancient rocks suggests that one decisive atmospheric rise, around 2.33 billion years ago, may have occurred within roughly 1 to 10 million years. To a cell dividing in hours or days, that interval is enormous. To a planet, it is almost sudden. Imagine compressing Earth's entire history into a single night. The planet forms at sunset. Oceans appear early in the evening. Life begins while the night is still young. For hour after hour, the atmosphere remains almost without oxygen. Photosynthetic microbes evolve, spread, and alter local waters, yet the wider sky continues in its ancient state. Then, late in the night, the chemistry shifts within the length of a few minutes. This comparison does not mean there was one exact instant when the world became oxygenated. The atmosphere probably moved through pulses, reversals, and regional differences. It means the final crossing may have been fast, compared with the time that came before. The reason lies in thresholds. For ages, oxygen production was matched by oxygen removal. Iron, sulfur, volcanic gases, methane, organic matter, and fresh rock absorbed the gas. As some sinks weakened and others became partly oxidized, each new oxygen molecule faced slightly less resistance. At first, the difference was tiny. Suppose the biosphere produced a vast amount of oxygen, but nearly all of it disappeared. If only a fraction remained, the atmosphere would still seem unchanged. Yet that remaining fraction could alter minerals and gases in ways that reduced future consumption. A little persistence made more persistence possible. Once oxygen stayed in the atmosphere, it began attacking methane more effectively. As methane declined, the atmospheric chemistry changed again. Ozone formed higher above the surface, modifying ultraviolet light. Weathering on land became more oxidative, releasing and trapping elements in new ways. Shallow waters received more oxygen from the air. Each consequence fed into other processes. The old balance had not simply shifted. The rules maintaining it were being rewritten. This is why planetary change can appear slow until it becomes difficult to stop. The atmosphere is not an empty container. It is a network of reactions. Move one part far enough, and other parts begin moving with it. Still, The oxygen concentration after the Great Oxidation Event was not close to the modern level. The sky did not suddenly become safe for humans. Estimates vary widely, but oxygen likely remained only a small fraction of present atmospheric abundance. Many deep ocean regions stayed anoxic. Methane did not vanish entirely. Anaerobic life continued in vast habitats. The revolution was incomplete, but it was irreversible in an important sense. Oxygen could rise and fall after the event, yet the atmosphere never returned permanently to its earlier condition of almost complete absence. The new gas had become part of Earth's long-term surface system. The distinction matters because popular stories often turn evolution into a staircase. First microbes appear, then oxygen rises, then complex cells form, then animals arrive. The real history is less orderly. Oxygen increased, fell, spread unevenly, and remained limited for long periods. Complex life required many later changes. The first rise opened possibilities, but it did not complete them. For the organisms already alive, even low oxygen could transform habitats. Shallow waters that once remained safe for anaerobes could become dangerous during more of the day and across wider regions. Oxidized minerals entered sediments. Metals changed their availability. New metabolic opportunities appeared beside new forms of stress. The transition also altered climate. Methane had helped warm the faint young Earth. As oxygen destroyed it, the planet may have lost part of its greenhouse protection. The same gas expanding biological possibilities could push the surface towards severe cold. This contradiction sits at the heart of the oxygen story. What appears beneficial from the viewpoint of modern animals was not beneficial to the world receiving it. The new atmosphere did not arrive as progress. It arrived as disturbance. No organism could see the global pattern. A cyanobacterial cell continued splitting water in sunlight. A methanogen continued producing methane in sediment. An anaerobic microbe encountered oxygen at the edge of its habitat and either survived or failed. The revolution emerged from these local events, multiplied until they became planetary. Geologists see the change through vanished sulfur signals, altered iron minerals, red beds, carbon isotope shifts, and the timing of glacial deposits. Each clue speaks a different language. Together they reveal an Earth crossing a threshold faster than its earlier stability would have suggested. The most unsettling part is not the speed alone. It is the lack of intention. No species chose to transform the atmosphere. No central force coordinated the event. Ordinary metabolism, repeated across enough time and space, became a planetary power. The old world had absorbed oxygen for ages. Then, within a narrow geological interval, it could no longer keep up. The sky gained a persistent new gas. The surface ocean began changing around it. The chemical boundaries that protected anaerobic communities moved inward and downward. From a modern distance, this can look like the beginning of a better Earth. But there were no animals waiting for breathable air, no forests ready to grow, and no future observers prepared to celebrate. There were only existing ecosystems facing a substance many of them could not tolerate. The threshold had been crossed. The atmosphere would never be exactly the same again. For much of life, the age of exposure had begun, and nothing alive had agreed to the change. Oxygen is easy to misunderstand because modern life has made peace with it. It is drawn into lungs, carried by blood, and used inside cells to release energy from food. Its presence feels gentle because the body surrounds it with layers of control. But remove those controls, and oxygen reveals another nature. It is highly reactive. It pulls electrons from other substances and can generate unstable molecules that attack proteins, membranes, and genetic material. Modern organisms produce enzymes and antioxidants that limit this damage. Ancient anaerobic cells, shaped in a world where free oxygen was rare, may have possessed far fewer defenses. As oxygen spread through shallow seas and coastlines, it did not need to reach modern concentrations to become dangerous. A small increase could interfere with sensitive enzymes. Repeated exposure could reduce growth, disrupt reproduction, and force communities away from habitats they had occupied for immense periods. The catastrophe would not have looked like later mass extinctions. There were no herds collapsing across plains, no forests burning beneath a darkened sky, and no shells disappearing suddenly from a rich fossil record. The victims were microscopic. Their bodies decayed quickly. The ocean floor on which many lived has long since been buried, heated, folded, or return to Earth's interior. What remains is a biological inference built upon a chemical transformation. If much of the surface biosphere depended on oxygen-poor conditions, then persistent oxygen must have destroyed or reduced many of those environments. Some lineages adapted, some moved, others almost certainly disappeared. Calling this Earth's first mass extinction therefore requires care. Scientists cannot produce a list of species lost or calculate a precise percentage. The term describes a likely global ecological crisis, not a fossil count equal to those available for later periods. The uncertainty should not be hidden, because the mystery becomes stronger when the limits of knowledge are clear. The rocks tell us the environment changed. Biology tells us that many organisms would have found the change lethal. Imagine a microbial mat near a shallow Archean shoreline. For generations beyond counting, its lower layers remain protected from oxygen. Sulfur-using microbes live beneath photosynthetic cells. Methanogens occupy deeper sediment. Nutrients move between the layers. The community is stable enough to seem permanent. Then daylight oxygen begins lasting longer. The upper boundary moves downward. Cells that once experienced exposure for minutes now face it for hours. Later, oxygen enters from the atmosphere even before local photosynthesis begins. The safe layers narrow. Organisms crowd into deeper mud, where food and light are scarce. There is no moment when the community understands that its world is ending. Each cell encounters only local chemistry. Yet over thousands of generations, populations shrink, boundaries shift, and familiar partnerships break apart. Some organisms produce enzymes that neutralize reactive oxygen. Those variants survive slightly better. Natural selection favors them. Others use the gas in new reactions, turning danger into opportunity. But adaptation has limits. Environmental change can move faster than beneficial traits spread. Small populations become vulnerable to chance. A lineage surviving in one basin may vanish from another. Oxygen also changes the surrounding minerals. Iron precipitates from water. Sulfur compounds take new forms. Metals previously locked in rock can become mobile, introducing additional toxins. Nutrients may be trapped or released. The catastrophe is not a single poison spreading through an unchanged sea. It is a complete reorganization of the chemical landscape. The old anaerobic biosphere had helped maintain methane-rich conditions. If methanogens declined in oxygen-exposed habitats, methane production could weaken further. Oxygen would face less opposition. The climate would cool. Habitat loss would then arise from both chemistry and temperature. This feedback gives the event its planetary character. Biological stress changes gas production. Gas changes climate. Climate changes ocean circulation, weathering, and nutrient supply. Those changes return to the biosphere. A local metabolic waste becomes part of a global crisis. Yet even this picture must avoid exaggeration. Anaerobic life did not disappear. Deep sediments remained protected. Hydrothermal systems continued supplying reduced compounds. Subsurface rocks contained water and chemical energy. Stagnant basins could remain oxygen-poor. The catastrophe redrew the map of habitability rather than replacing every environment at once. Its survivors are still present. Methanogens live in wetlands and digestive systems. Sulfate-reducing microbes occupy mud beneath oxygenated water. Anaerobic organisms persist in deep crustal fractures and oxygen-starved basins. They are not relics frozen in time, but evolving descendants of lineages that learned to endure a planet no longer organized around them. The extinction story therefore contains an uncomfortable reversal. From the viewpoint of oxygen-sensitive life, cyanobacteria were not humble builders of a better world. They were agents of contamination. Their success reduced the space available to others. The atmosphere celebrated in modern textbooks was a spreading environmental hazard. This does not make cyanobacteria villains. They had no intention, and intention is not required for consequence. They followed the logic of survival, using sunlight and water more effectively than many competitors. Their waste accumulated because the planet changed around them. The same principle appears throughout Earth's history. Life alters its environment. The alteration creates winners and losers. A successful metabolism can destabilize the conditions that allowed it to emerge. Evolution has no promise of balance. As oxygen became permanent, the old world did not end everywhere. It fractured. Safe regions became isolated. boundaries grew sharper. Near the surface, organisms faced the most reactive atmosphere Earth had yet known. Deeper below, anaerobic communities continued as though hidden beneath the future. The first great oxygen crisis was therefore both extinction and migration. Life withdrew into refuges, adapted along chemical borders, and explored reactions that had once been impossible. The next stage belongs to those survivors. Some escaped the poison, some learned to defend themselves. A few began to use oxygen's hunger for electrons as a new source of power. In that shift, catastrophe became selection, and selection began building possibilities no earlier biosphere could have predicted. This weapon would become life's hidden engine. The spread of oxygen did not divide life neatly into survivors and victims. Between extinction and safety lay a wide range of imperfect responses. Some organisms retreated, some endured brief exposure, some repaired damage after it occurred. Others found ways to use the new gas without allowing it to destroy them. The deepest refuges were physical. Mud beneath the seafloor remained oxygen poor because oxygen entering from above was consumed near the surface. Cracks within rock offered water, minerals, and protection from the atmosphere. Hydrothermal systems released reduced chemicals that quickly removed oxygen. In stagnant waters, weak circulation allowed deeper layers to remain anoxic even when the surface changed. Life withdrew into these places, though withdrawal is only a human description. Individual cells did not travel because they understood a planetary danger. Populations already living in protected regions continued reproducing, while those in exposed regions declined. Over time, the map of anaerobic life shifted away from sunlit surfaces toward hidden environments. That hidden world still exists. Beneath wetlands, methane-producing microbes occupy sediments beyond the reach of oxygen. Inside the intestines of animals, descendants of ancient anaerobes live in warm darkness. Under the seafloor, cells survive at extraordinary depths and move through time at rates far slower than surface life. The oxygen revolution did not erase the old biosphere, it buried much of it. At the boundary, evolution followed another path. Small variations in cellular chemistry could determine survival. An organism able to repair an oxidized protein lived longer. A cell producing an enzyme that converted a dangerous oxygen molecule into a less harmful form gained an advantage. Protective pigments, metal-binding proteins, and improved membrane control became increasingly useful. These defenses did not appear all at once. Many may have existed earlier for other reasons. Reactive molecules can arise even in low oxygen environments, through sunlight, minerals, or internal metabolism. Evolution often rebuilds old tools for new crises. A protein that once handled one kind of chemical stress may be modified to manage oxygen. Gene exchange also mattered. Microbes can acquire useful genes from distant relatives, rather than waiting for every solution to evolve independently. A protective enzyme arising in one lineage may spread through a community. The oxygen crisis therefore encouraged not only competition, but the movement of survival tools across the microbial world. Some organisms went beyond defense. They began using oxygen as part of reactions that released energy. Oxygen accepts electrons eagerly. That property makes it dangerous, but it also creates a powerful metabolic opportunity. If a cell controls the transfer, the same chemical force that attacks its structures can help power its life. At first, oxygen-using metabolisms may have operated only where the gas appeared temporarily. A microbe near the top of a mat could use oxygen during daylight, then switch to another strategy at night. Another might remain at the moving boundary between oxygenated and anoxic water. Flexibility became valuable in a world that changed across hours, seasons, and depths. The new chemical gradients created niches that had never existed. Where oxygen met methane, certain microbes could gain energy by consuming the methane. Where oxygen reached iron or sulfur compounds, other organisms could exploit the reactions. Every boundary was both a danger zone and a source of power. This is one reason the catastrophe cannot be understood only as destruction. Oxygen closed large areas to sensitive organisms, yet it also multiplied the number of possible metabolisms. The old world had offered rich chemical diversity, but the addition of a highly reactive gas created entirely new combinations. Minerals changed as well. Oxidative weathering released elements from continental rocks. Some nutrients entered rivers and shallow seas. Other elements became trapped in insoluble minerals. Toxic metals could become more mobile. Life faced a surface environment that was richer in some ways and harsher in others. Natural selection did not move toward complexity with a clear destination. It responded to each local problem. A membrane became slightly more resistant. A repair system became faster. A metabolic pathway captured more energy. A community arranged itself around a new boundary. Across millions of years, these small changes accumulated into a biosphere increasingly capable of living with oxygen. The contrast with the earliest oxygen exposure is striking. At first, the gas was an unwanted waste entering ecosystems built without it. Later, entire communities depended on its presence. What had begun as contamination became part of the structure of life. But the conversion was incomplete. Oxygenated surface waters existed above anoxic depths. Anaerobes survived beside aerobes. Some organisms required oxygen, others tolerated it, and others still died upon contact. The planet became chemically divided. These divisions produced ecological movement. Organic matter created at the surface sank into deeper water, feeding anaerobic communities. Reduced compounds rose from sediments and encountered oxygen above. Microbes positioned themselves along the exchange. The biosphere became a layered engine, connecting two incompatible worlds. The survivors therefore did more than endure. They transformed the catastrophe into a source of diversity. Every defense opened a new habitat. Every new metabolism changed the products available to other organisms. Evolution began exploring a larger chemical space. Yet oxygen was altering more than cells and minerals. It was reaching the atmosphere, where methane had helped trap heat beneath the faint young sun. As oxygen increased, methane became harder to maintain. The greenhouse shield weakened, and a planet already receiving less sunlight than today began losing warmth. The oxygen crisis was about to change form. What began as a poison in shallow water would become a force acting on the global climate. The survivors had learned to live beside oxygen, but they would now face a danger no enzyme could neutralize: ice advancing across the world. The new atmosphere had selected its first winners, but winning offered no safety. Their success helped strengthen the gas that threatened methane, while every altered mineral and buried fragment of carbon pushed the planet farther from its former balance. Adaptation had solved one crisis and prepared another. The early sun was weaker than the sun shining over Earth today. Its light carried less energy, yet ancient rocks show that liquid water remained on the surface. Rivers flowed, sediments settled in oceans. Life spread through environments that could not have existed on a permanently frozen planet. Something kept the young Earth warm. Carbon dioxide contributed to the greenhouse effect, trapping part of the heat that would otherwise escape into space. Water vapor added more warming. But methane, released in large quantities by anaerobic microbes, may have been one of the atmosphere's most important blankets. Methane is powerful because it absorbs heat efficiently. Even at lower concentrations than carbon dioxide, it can strongly influence climate. In an oxygen-poor atmosphere, methane also survives longer than it does today. Methanogens living in sediments and shallow seas could continually replace what was destroyed by sunlight and other reactions. The result may have been a hazy, methane-supported world warm enough for open oceans beneath the faint young sun. Then oxygen began to persist. Oxygen and methane cannot remain together indefinitely. Their chemistry drives them toward carbon dioxide and water. In the modern atmosphere, methane survives only because biological and geological sources continually add more. As oxygen increased during the Great Oxidation Event, methane faced a much stronger route toward destruction. At the same time, methane production may have weakened. Nickel limitation could have reduced the activity of methanogens in some regions. Oxygen entering shallow habitats could have pushed methane producers into deeper sediments. New microbes may have consumed methane where it met oxygen. Several pressures acted together. The atmospheric blanket began thinning. Carbon dioxide remained, but replacing methane with carbon dioxide does not preserve the same amount of warming molecule for molecule. The planet was already receiving reduced sunlight. A major loss of methane could therefore push temperatures downward. The rocks record severe glaciations during the Paleoproterozoic era. broadly overlapping the oxygen revolution. These glacial intervals are often grouped under the name Huronian glaciation. Ancient deposits contain stones carried by ice, scratched surfaces shaped beneath glaciers, and sediments laid down in cold environments. Some evidence suggests ice reached low latitudes, though the exact extent and timing of each glaciation remain debated. The connection appears almost too dramatic. Microbes release oxygen, oxygen destroys methane, the greenhouse weakens, ice advances across the planet. But Earth rarely follows a single chain without complication. Continents were moving and growing. Their positions affected ocean currents and the amount of sunlight reflected by land and ice. Weathering removed carbon dioxide from the atmosphere. Volcanic activity returned carbon dioxide and other gases. Biological productivity changed the burial of carbon. Each process influenced climate. The methane collapse is therefore a leading part of the explanation, not a solitary cause carved beyond doubt. Still, the timing invites attention. Oxygen's rise altered exactly the gas capable of warming a faint young Earth. The same biological innovation that opened new chemical opportunities may have helped create one of the most severe climate crises in the planet's history. Imagine the change unfolding slowly from the viewpoint of life. Summers become cooler, seasonal ice remains longer. Glaciers grow on high ground, then move toward lower regions. White surfaces reflect sunlight that dark rock or open water would have absorbed. Cooling creates more ice, and more ice creates further cooling. This is the ice albedo feedback. It can turn a modest decline in temperature into a much larger transformation. Once glaciers expand far enough, the planet reflects more solar energy to space and becomes harder to warm. Oceans begin freezing at the surface. Circulation changes. Nutrient supplies shift. Sunlit habitats narrow. Microbial communities adapted to warm, shallow seas encounter cold, darkness, and physical isolation. The oxygen catastrophe has become a climate catastrophe. The irony deepens. Methanogens helped warm the world in which oxygen-producing microbes evolved. Their atmospheric product supported liquid water and widespread habitats. As oxygen weakened methane, it attacked part of the environmental foundation shared by both groups. Cyanobacteria could survive cold conditions, but photosynthesis depends on light and liquid water. Expanding ice reduced both. Oxygen production itself may have fallen as habitats froze. The process that triggered cooling could therefore weaken during the crisis, creating another feedback whose direction changed through time. Beneath the ice, anaerobic life continued. Volcanic regions remained active. Hydrothermal vents supplied heat and chemicals. Open water may have persisted near the equator, along coastlines, or in areas of thin seasonal ice. The planet was never chemically or physically uniform. Whether the Huronian episodes created a complete snowball Earth remains uncertain. Some models allow thick global ice. Others favor open ocean regions or less extreme glaciation. The ancient record is incomplete, and several separate ice ages may have occurred rather than one continuous freeze. For the story, the important truth does not require a perfectly sealed snowball. Earth experienced glaciation severe enough to reshape continents, oceans, and biological habitats during the same broad interval in which oxygen transformed the atmosphere. Life had engineered a climate it had never faced before. No cell intended to cool the planet. No organism understood methane as a greenhouse gas. The outcome emerged from the interaction of metabolism and atmospheric chemistry. A waste product released in shallow water traveled through a chain of reactions until glaciers moved across stone. This is the hidden scale of biological power. Life does not need intelligence to alter a planet. It needs abundance, time, and a metabolism connected to the atmosphere. The cooling world now approached another threshold. Ice spread, sunlight reflected away, and weathering slowed beneath frozen surfaces. Yet volcanoes continued releasing carbon dioxide. If that gas accumulated for long enough, warming would eventually return. The freeze was not only an ending. Beneath its quiet surface, Earth was preparing the conditions for thaw, erosion, nutrient release, and another surge in the oxygen story. The gas that would one day sustain animals had become a force of winter, proving that habitability depends not on one substance, but balance. Ice changes a planet quietly. It does not need fire, impact, or thunder. It advances by remaining where yesterday's snow would have melted. A thin white cover becomes a field. A field becomes a glacier. The glacier moves under its own weight, grinding rock, carrying debris, and pressing the memory of cold into the land beneath it. During the Paleoproterozoic glaciations, vast regions of Earth may have entered this slow transformation. Continental surfaces that had once received rain and sunlight became covered by ice. Shallow seas froze seasonally or for longer periods. Ocean circulation weakened or reorganized. Light-reaching photosynthetic communities declined. The exact appearance of the planet remains uncertain. Some researchers have considered conditions approaching a global snowball Earth, with ice spreading to very low latitudes. Others argue that open water survived across significant regions. The ancient deposits record severe cold, but they do not provide a complete photograph of every ocean. For life, the difference between total and nearly total ice mattered enormously. Yet survival did not require the planet to remain comfortable. It required only enough refuges. Open water near volcanic islands could absorb sunlight. Hydrothermal vents warmed surrounding seawater from below. Thin ice might allow light to pass through. Coastal cracks, seasonal melt zones, and equatorial regions could preserve liquid habitats. Beneath glaciers, water moved along rock under pressure. Deep sediments remained insulated from the cold above. Microbial life is skilled at finding narrow possibilities. Modern organisms live within ice, beneath glaciers, in briny channels, and around vents where temperature and chemistry change over short distances. Their ancient relatives were not identical, but they had already survived billions of years of environmental variation. The frozen planet would still have been alive. In sunlit refuges, cyanobacteria continued photosynthesis when conditions allowed. Anaerobic microbes remained active below the reach of oxygen. Methanogens produced gas in sediments. Other organisms used sulfur, iron, and hydrogen. Biological activity slowed in many places, but it did not stop everywhere. The ice also changed how Earth interacted with sunlight. White surfaces reflected much more energy than dark ocean or bare rock. As ice expanded, the planet absorbed less heat. Temperatures fell further, allowing more ice to form. This feedback made the cold state difficult to escape. Yet another process moved in the opposite direction. Volcanoes continued releasing carbon dioxide. On a warm, wet planet, chemical weathering removes carbon dioxide over long periods, as rain reacts with silicate rock and carries dissolved products toward the ocean. Under widespread ice, much of that weathering slows. Carbon dioxide begins accumulating. The buildup may take millions of years. Nothing in the frozen landscape seems to change. Volcanoes release gas into a pale sky. Ice continues reflecting sunlight. Then the greenhouse effect becomes strong enough to weaken the cold state. Melting exposes dark surfaces, which absorb more sunlight. Retreat creates more retreat. The thaw can be as self-reinforcing as the freeze. Glaciers leave behind more than water. Their movement crushes and scrapes rock into fine material. When melting begins, rivers carry this fresh mineral powder into lakes and seas. Nutrients that were locked in stone become available to microbes. Iron, phosphorus, and other elements enter coastal waters in new quantities. This release may stimulate biological productivity. Cyanobacteria spread through newly opened water. Organic matter forms and sinks. This is not a simple reward built into nature. Glacial erosion can also expose reduced minerals that consume oxygen. Nutrient release can produce blooms followed by decay, drawing oxygen from water. Different effects dominate in different places and at different times. But the freeze rearranged the surface system so completely that the thaw could not return Earth to its previous condition. Continents emerged from the ice scraped and chemically fresh. Oceans received pulses of sediment. Atmospheric carbon dioxide had increased. Methane and oxygen continued their uneasy competition. The biosphere expanded into habitats that had been closed. For a late-night listener, it is tempting to imagine the thaw as a sunrise after endless winter. In geological reality, it unfolded across ages. Ice margins shifted, seas opened and closed. Glaciers returned during later cold intervals. The Huronian record may contain several major glaciations separated by warmer periods. Each cycle altered the relationship among climate, weathering, nutrients, and oxygen. The planet was learning no lesson. It was following feedbacks, Yet from those feedbacks came a pattern that resembles trial and consequence. Life changed the atmosphere. The atmosphere changed climate. Climate reshaped the continents. The continents fed new material back to life. No part of the system remained separate. Beneath the frozen sky, organisms also faced isolation. Populations separated by ice and changing seas could evolve along different paths. Refuges became biological islands. When conditions improved, surviving lineages expanded into emptied or altered habitats. Extinction and diversification followed one another, though the microbial fossil record hides most of the details. The quietest places may have mattered most. In deep mud, communities continued chemical cycles without sunlight. In cracks beneath the crust, water and rock supplied energy. These refuges preserved old metabolisms, while surface environments moved toward oxygen. They ensured that the planet would remain a mixture of ancient and new chemistry. When the ice retreated, the oceans did not become modern. Deep waters remained largely anoxic. Oxygen existed unevenly. The atmosphere held more of it than before, but still far less than today. The thaw created opportunity, not completion. Soon the rocks began recording an extraordinary change in carbon. For more than 100 million years, carbonate sediments across several continents carried unusually heavy carbon isotope values. This event, known as the Loma-Gundi excursion, may signal immense burial of organic matter and a temporary excess of oxygen production over consumption. The Frozen Age had not ended the oxygen revolution. It may have loaded the next stage. As rivers carried glacial debris into warming seas, the planet approached another unstable abundance. After surviving poison and ice, life was about to experience an oxygen overshoot, whose rise and collapse would shape the next billion years. After the ice began to retreat, Earth's rocks recorded a change so large that it remains one of the most striking chemical events in the planet's history. Across carbonate formations separated by oceans and continents, the ratio of carbon isotopes shifted toward unusually heavy values. The disturbance lasted for more than 100 million years. This is the Loma-Gundi event. Its name comes from regions where the signal was first studied, but the pattern is global enough to suggest a major reorganization of the carbon cycle. To understand why it matters, the story returns to photosynthesis. Oxygen-producing organisms use carbon dioxide and water to build organic matter. In the process, they release oxygen. Most organic matter is eventually consumed. Microbes break it down, carbon returns to water or air, and the oxygen connected with its creation is used again. The planetary account closes. But if organic matter is buried before it decays, the account remains open. Carbon is locked into sediment, and a corresponding amount of oxygen can remain in the surface system. The Loma-Gundi isotope pattern has often been interpreted as evidence that unusually large quantities of organic carbon were buried. If that interpretation is correct, oxygen production may have greatly exceeded oxygen consumption for a long interval. The atmosphere and parts of the ocean could have become more oxygenated than they had ever been before. Imagine sediments as a vault. Every buried fragment of biological carbon seals away material that would otherwise react with oxygen. Layer upon layer accumulates beneath the seafloor. The living surface continues making organic matter, while the buried portion no longer participates in the immediate cycle. Oxygen is left outside the vault. Across millions of years, even a small imbalance becomes enormous. Some researchers have suggested that atmospheric oxygen during the Loma-Gundi interval may have approached a significant fraction of modern levels. Others argue for much lower values. The available chemical indicators do not agree perfectly, and models depend on uncertain assumptions about volcanic gases, ocean chemistry, weathering, and how quickly carbon moved between reservoirs. The disagreement is important. It prevents the event from becoming a simple claim that Earth briefly gained modern air 2 billion years ago. What the rocks support more clearly is an exceptional disturbance involving carbon burial and expanded oxidation. The exact amount of free oxygen remains debated. The source of the burial is also uncertain. Nutrient-rich runoff after glaciation may have stimulated photosynthesis. Growing continents and changing seas may have created broad, shallow environments where organic material could accumulate. Phosphorus, nitrogen, iron, and trace metals influenced productivity. Tectonic basins buried sediments at different rates. No single mechanism explains every location. The event may also contain several phases rather than one uniform global condition. Some oceans could have been oxygenated near the surface, while deeper waters remained anoxic. Productive coastal regions may have buried large amounts of carbon, even as other basins stayed chemically limited. Again, the planet resisted simplicity. Still, the possibility of an oxygen overshoot changes the narrative. The Great Oxidation Event is often pictured as the first step of a steady climb toward the present. The Lomagundi record suggests something more unstable. Oxygen may have risen beyond a sustainable balance, remained elevated for tens of millions of years, then fallen. The first atmospheric revolution may have overcorrected. During this interval, oxidative weathering on land intensified. Minerals exposed to air and rain reacted with oxygen. Sulfide minerals broke down, sending sulfate and metals toward the sea. The oceans gained new nutrients and new toxins. Microbes adapted to oxygen found expanding habitats, while anaerobic organisms retreated further from exposed environments. In Gabon, rocks around 2.1 billion years old preserve controversial structures that some scientists interpret as early multicellular or colonial organisms. Their age overlaps the broader world, created after oxygen's rise and during the later part of the Lomagundi interval. If these structures are biological, they may represent life taking advantage of a temporary oxygen-rich window. Complexity appeared, explored a new possibility, and then vanished when conditions changed. That connection remains uncertain, but the timing is difficult to ignore. An overshoot also contains the conditions of its own ending. Large-scale burial of organic carbon cannot continue forever without changing nutrient supplies and the availability of places where burial occurs. Continents erode, basins fill. Previously buried material is uplifted and exposed. Oxygen reacts with reduced minerals and old organic deposits. The planetary account begins closing again. As oxygen rises, it strengthens the processes that consume oxygen. Weathering reaches more reduced rock. Sulfate enters the sea and supports microbial metabolisms that break down organic matter. Newly oxygenated habitats allow respiration to use carbon more efficiently. The surplus creates demands that can eventually reduce the surplus. This does not mean the Lomagundi event was a perfectly self-correcting cycle. Earth's carbon system is too complex for that. But it shows why an oxygen increase can be temporary even after the atmosphere has crossed into permanent oxygenation. The most haunting evidence comes from what follows. After the great positive carbon isotope excursion ends, records suggest a major decline in biological productivity and oxygen availability. The biosphere that had expanded into new chemical space may have contracted. The planet did not continue climbing toward complex life. It entered a long interval of lower oxygen, restricted nutrients, and slow visible change. The Lomagundi puzzle therefore stands between two worlds. Behind it lies the first oxygen catastrophe and severe glaciation. Ahead lies a biological slowdown so prolonged that later scientists would call the middle Proterozoic boring. But nothing about the transition was truly boring. The rocks suggest a planet experimenting with abundance, pushing oxygen higher than before, then losing much of that gain. For a brief geological age, Earth's surface may have approached conditions capable of supporting forms of life far more organized than the ancient microbial mats. Then the window narrowed. The mystery is not only why oxygen rose, it is why the planet could not keep it there. When the Lomagundi carbon isotope signal ended, the planet did not settle into a stable oxygen-rich future. Instead, the geological record suggests a profound reduction in biological activity. The change is difficult to see because it is written not in the disappearance of large creatures, but in the weakening of chemical signals produced by life. Researchers studying carbon and sulfur isotopes have argued that global primary productivity fell dramatically after the oxygen overshoot. Primary productivity is the creation of organic matter from carbon dioxide, mainly through photosynthesis. It forms the base of nearly every ecosystem. A sustained decline would mean less biological material entering the oceans, less energy moving through food webs, and less oxygen being released. Some estimates describe this as one of the largest long-term reductions in the productivity of Earth's biosphere. The exact magnitude remains uncertain, but the direction appears significant. Why would a world that had discovered oxygen-producing photosynthesis suddenly become less productive? The answer may lie in nutrients and feedbacks. Photosynthetic organisms need more than light, water, and carbon dioxide. They require phosphorus, nitrogen, iron, and other elements. If those nutrients become trapped in sediments or altered minerals, biological growth slows. Oxygen can encourage the formation of iron oxides that bind phosphorus and carry it to the seafloor. A more oxygenated ocean may therefore remove part of the nutrient supply needed to produce more oxygen. The revolution begins limiting itself. Large quantities of organic carbon buried during the Lomagundi interval may later have been exposed through erosion or tectonic uplift. When that old carbon reacted with oxygen, it consumed part of the atmospheric surplus. Reduced minerals brought to the surface did the same. Volcanic gases continued adding substances that drew oxygen down. Meanwhile, changes in ocean circulation and continental arrangement affected where nutrients entered the sea. Productive shallow environments could shrink. The basins that had buried carbon efficiently could fill or become less favorable. The imbalance that raised oxygen weakened. Atmospheric oxygen fell, though it did not return to its earliest near-zero state. Surface waters remained oxygenated in many regions. Deeper oceans, however, were often anoxic. Some areas became rich in dissolved sulfide, creating conditions hostile to organisms needing oxygen and to many forms of nutrient cycling. The ocean became layered. At the top, sunlight supported photosynthesis. Oxygen entered from the atmosphere and local production. Below, decomposition consumed the gas faster than circulation replaced it. Deeper still, anaerobic microbes used sulfate and produced sulfide. Boundaries shifted with climate, seasons, and ocean circulation. This arrangement could persist for immense spans of time. Oxygen existed, but it did not reach every place where complex life might benefit from it. Nutrients existed, but their distribution limited productivity. The planet had crossed the first great threshold without escaping the chemical controls that kept complexity rare. This long interval is often associated with the so-called boring billion, a broad stretch of the Proterozoic, during which atmospheric composition, climate, and visible evolution appeared relatively stable compared with the dramatic events before and after. The name is memorable, but misleading. Nothing living through that time would have experienced boredom. Microbes competed across oxygen boundaries. Early eukaryotic cells emerged and diversified. Continents assembled and broke apart. Oceans shifted between anoxic, sulfidic, and locally oxygenated states. The apparent calm comes from the limited record and the absence of large, easily fossilized organisms. The more important lesson is delay. Oxygen had risen, yet animals did not appear immediately. A gas necessary for large active bodies was present, but not at modern abundance, and not evenly distributed. Complex cells required additional innovations. Ecosystems needed stable nutrient cycles, favorable habitats, and time. This delay overturns the idea that oxygen alone commanded evolution to become complex. Oxygen expanded the range of possible life, but possibility is not destiny. The slowdown may also have eliminated early experiments. If organized colonies or multicellular forms appeared during the oxygen overshoot, falling oxygen and productivity could have removed the environments supporting them. Their disappearance would leave only uncertain structures in rare rocks. A planet can open a door, then close it before anything passes through permanently. The biological decline also changed the atmosphere. Less photosynthesis meant less oxygen production. Less organic matter meant less burial. Lower oxygen allowed methane to survive more easily, though the atmosphere did not fully return to its Archean condition. Climate and chemistry found a new balance, one quieter than the crisis that created it. For hundreds of millions of years, Earth remained between worlds. It was no longer the anaerobic planet ruled entirely by methane producers and oxygen-sensitive communities. It was not yet the deeply oxygenated planet capable of supporting active animals throughout the ocean. The surface carried a partial future above ancient depths. This in-between state matters, because evolution often works within constraints, rather than waiting for ideal conditions. Organisms learned to use limited oxygen. Eukaryotic cells developed internal structures. Predation and cooperation emerged at microscopic scales. Genetic systems expanded. The foundations of complexity formed slowly, while the planet appeared still. The largest biological slowdown no one saw was therefore not an empty age. It was a period of hidden preparation. The oxygen catastrophe had removed old habitats, created new metabolisms, changed climate, and produced an overshoot. Its aftermath imposed limits. Those limits shaped which innovations survived and how quickly they spread. Soon the story must leave the atmosphere and enter the cell. Oxygen's greatest influence was not simply that it accumulated above the ocean. It was that organisms learned to guide its reactivity through controlled pathways, capturing energy that older metabolisms could not access as efficiently. The planet possessed oxygen, but not enough everywhere to unlock every outcome. Inside certain cells, however, even a limited supply could be transformed into power. The next revolution would not first appear in the sky. It would unfold across a membrane, where electrons moved in silence, and the poison of the ancient world became a source of organized energy for complex life. Oxygen did not become useful simply because it entered the atmosphere. Before a cell could benefit from the gas, it had to control a reaction powerful enough to destroy it. The difference between poison and energy was organization. When organic molecules react freely with oxygen, energy is released as heat. Fire is the visible form of that process. A cell cannot survive by burning its food in a sudden, uncontrolled reaction. It needs to release energy through small steps, capturing part of it in chemical forms that can be used for movement, repair, growth, and reproduction. Aerobic respiration performs this controlled burning without flame. The process begins by removing electrons from food molecules. Those electrons travel through a chain of proteins embedded in a membrane. At each step, a small amount of energy is used to move charged particles across the membrane, creating a difference between the two sides. The cell then allows the particles to flow back through another molecular machine, which produces a portable energy-carrying molecule. Oxygen waits at the end of the chain. It accepts the electrons and combines with hydrogen to form water. Without that final receiver, the flow slows or stops. The complete chemistry is intricate, but its evolutionary meaning is simple. Oxygen allows cells to extract much more usable energy from many kinds of organic matter than common fermentation pathways can. It turns a highly reactive gas into the final step of a carefully governed system. That advantage comes with danger. Electrons do not always follow the intended route. Some escape and react with oxygen prematurely, producing unstable molecules. These reactive forms can attack DNA, proteins, and membranes. Aerobic life therefore requires both energy machinery and protection. Cells evolved enzymes that convert dangerous oxygen products into safer substances. They developed repair systems for damaged molecules. Antioxidant compounds absorbed part of the chemical stress. Membranes and metal-handling proteins reduced unwanted reactions. The price of using oxygen was constant maintenance. This reveals a deeper truth about evolutionary innovation. New power rarely arrives without new vulnerability. Oxygen supported more efficient metabolism, but it also created kinds of cellular damage that had barely existed in the earlier world. The first organisms to use oxygen may not have depended on it completely. In habitats where the gas appeared only during daylight, flexibility mattered. A cell might use aerobic reactions when oxygen was available, then switch to fermentation or another electron acceptor when darkness returned. Over time, lineages living in more consistently oxygenated environments could specialize. The energy advantage changed competition. An organism extracting more energy from the same food could grow faster, repair damage more effectively, or survive periods of scarcity. It might explore habitats unavailable to strict anaerobes. Yet oxygen use did not make every aerobic organism superior in every setting. In oxygen-poor mud, the old metabolisms remained better suited. Evolution filled both worlds. Oxygen also expanded the chemistry cells could build. Certain reactions used in the production of sterols and related molecules require oxygen. Sterols help regulate membranes in many eukaryotic cells, affecting flexibility, signaling, and internal organization. Parts of this biochemical machinery appear to have ancient bacterial origins, suggesting that microbes began exploring oxygen-dependent construction long before animals existed. The gas was becoming more than a way to release energy. It became a tool for manufacturing new cellular components. Still, oxygen alone did not create complex cells. Many bacteria use aerobic respiration and remain small. Some eukaryotes can survive with little or no oxygen. Cell complexity depends on genetic regulation, internal membranes, structural proteins, communication systems, and ecological pressures. Energy is part of the story, not a complete explanation. But energy sets boundaries. A cell must pay for every gene it copies, every protein it builds, every membrane it maintains, and every mistake it repairs. More available energy can support systems that would otherwise be too expensive. This is why the relationship between oxygen and complexity remains so compelling. The Great Oxidation Event did not immediately produce large organisms, but it created expanding environments in which energy-rich metabolism was possible. It changed the budget available to evolution. For hundreds of millions of years, microbes tested that budget. Some used oxygen at the edges of mats. Some lived in shallow waters where the gas entered from the atmosphere. Others consumed organic matter sinking from photosynthetic zones. Their metabolisms linked surface oxygen to deep carbon cycles. The chemical boundaries of the ocean became living engines. Oxygen moved downward. Reduced compounds moved upward. Cells gathered where the two met and captured energy from the encounter. Life increasingly depended on differences created by the atmospheric revolution. Inside these organisms, oxygen's double nature never disappeared. The gas powered metabolism while threatening the machinery performing it. Modern aerobic cells preserve the same tension. They require oxygen and suffer when its reactions escape control. Every breath taken by an animal, therefore, carries an ancient compromise. Oxygen is admitted, transported and delivered, but never allowed to wander freely. Proteins bind it. Membranes guide its use. Enzymes manage the damage. Life survives by keeping the old poison on a molecular leash. At some point in the Proterozoic, one cell entered a lasting relationship with an oxygen using bacterium. The bacterium did not remain an independent guest. Across evolutionary time, it became an organelle. That organelle was the mitochondrion. The partnership would reshape energy production inside complex cells, but it began as an encounter between organisms whose ancestors had survived opposite sides of the oxygen crisis. A host adapted to one set of conditions, joined with a bacterium skilled at using the gas that had transformed the planet. Competition had driven much of the earlier story. Before that alliance, oxygen had changed the outer world. After it, the same chemistry would be organized inside a cell, concentrated behind membranes, and inherited by descendants that would eventually include algae, fungi, plants, animals, and human beings. The ancient waste product had become controlled fire. Soon it would find a permanent home within complexity. The origin of the complex cell is not preserved as a single fossil. No rock contains the moment when one organism entered another and remained there. The evidence lives inside cells that exist now. Every animal, plant, fungus, and many single-celled eukaryotes contain mitochondria or structures descended from them. Mitochondria possess their own small genomes. They divide in a manner resembling bacteria. Their membranes, ribosomes, and genes point toward an ancient bacterial ancestor. The conclusion is difficult to escape. Mitochondria began as free living cells. At some point, an ancestral host formed a relationship with one of those bacteria. The exact nature of the first encounter is unknown. The host may have swallowed the bacterium as food and failed to digest it. The bacterium may have entered as a parasite. Whatever began the relationship, neither partner remained unchanged. The bacterium lived inside the host, where it gained protection and a steady supply of material. The host gained access to the bacterium's metabolism. Genes moved from the bacterial genome into the host genome. Proteins that once functioned in an independent organism became parts of a shared cellular system. Across time, the partnership became impossible to separate. The bacterium became the mitochondrion. This event, called endosymbiosis, is one of the deepest mergers in evolutionary history. It did not create a temporary alliance like two species living beside one another. It created a new level of organism in which former partners became inherited components of the same cell. Mitochondria are often introduced as powerhouses, but that phrase captures only part of their importance. They help generate cellular energy, regulate metabolism, influence signaling, manage calcium, participate in programmed cell death, and interact with the cell's response to stress. Their membranes create controlled spaces where reactions can be arranged with great precision. The energy question remains central. A bacterium living inside another cell brought many small energy-producing membranes into the host's interior. As the number of mitochondria increased, the cell could potentially support a larger genome, more proteins, internal transport, and complex regulation. Some researchers argue that this energy surplus was essential for eukaryotic complexity. Others caution that the sequence is not so simple. The host may already have possessed complex features before acquiring mitochondria. Many bacteria have extensive internal membranes and sophisticated behavior without becoming eukaryotes. Energy availability alone cannot explain nuclei, chromosomes, cytoskeletons, sexual reproduction, and the many other traits of complex cells. The debate does not weaken the importance of the merger. It changes the question from whether mitochondria mattered to exactly how and when they mattered. Dating the event is difficult. Molecular clocks compare genetic differences among living organisms to estimate when their ancestors separated, but rates of change vary. Fossils of early eukaryotes are rare and often disputed. Some evidence places important stages of eukaryotic diversification deep in the Proterozoic, long after the first atmospheric oxygen rise. This gap matters. The Great Oxidation Event did not immediately produce mitochondria, eukaryotes, or animals. It created a new planetary setting in which oxygen-using bacteria could expand, and oxygen-dependent metabolisms could become more valuable. The merger required additional evolutionary accidents and ecological opportunities. The connection is therefore indirect but profound. Without oxygenic photosynthesis, widespread aerobic respiration would not have become possible. Without aerobic bacteria, the mitochondrial ancestor would not have possessed the same metabolic role. Without endosymbiosis, the later history of complex life would have followed a path difficult to imagine. The alliance joined descendants of the oxygen crisis. The host belonged to a world still shaped by anaerobic chemistry. The bacterial partner had learned to manage the reactive gas. Inside one cell, two evolutionary histories met. At first, the relationship may have been unstable. The bacterium could reproduce too quickly and harm the host. The host could digest or expel it. Their metabolisms had to become coordinated. Gene transfer reduced the bacterium's independence. Protein transport systems evolved so that instructions stored in the host genome could send products back into the mitochondrion. Generation after generation, conflict became dependence. This pattern appears elsewhere in evolution. Chloroplasts inside plants and algae descend from cyanobacteria captured by a eukaryotic cell. The organisms that once released oxygen into ancient seas later became the internal structures performing photosynthesis in leaves. Planetary change became cellular inheritance twice, first through the atmosphere, then through symbiosis. The mitochondrial partnership opened possibilities for larger cells with more internal organization. Membranes divided tasks into separate compartments. A nucleus protected and managed genetic material. A cytoskeleton supported shape, movement, and transport. Cells could engulf particles, communicate, and in some lineages, join into colonies. None of these outcomes was inevitable. Most eukaryotic lineages remained microscopic. Many experiments ended. Environmental oxygen fluctuated, nutrients remained limited, and deep oceans stayed hostile. But the range of possible organisms had expanded again. The Oxygen Catastrophe had once forced life apart, pushing anaerobes into refuges and favoring organisms capable of managing the gas. Endosymbiosis reversed that pattern. Different metabolisms were brought together inside a shared boundary. What began as environmental poison became an internal partnership. The rocks should, in principle, contain traces of the new complexity. Yet early eukaryotes were soft, small, and difficult to recognize. Their fossils can resemble mineral structures or simple colonies. Chemical markers offer clues, but contamination and later alteration complicate interpretation. Then, in rocks from Gabon, dating to about 2.1 billion years ago, researchers found structures far larger and more organized than ordinary microbes. Some appear disc-shaped, lobed, or elongated. Their discoverers argue that they may represent colonial or multicellular life, living in oxygenated waters after the Great Oxidation Event. Other scientists remain unconvinced. The debate leads toward a haunting possibility. Complex organization may have emerged during an early oxygen-rich window, only to vanish when conditions declined. Evolution may have opened the door to larger life, stepped through briefly, and then been forced back. The alliance that built complex cells did not guarantee a lasting complex world. The planet still had to become ready to support one. In the forests of Gabon, beneath layers of younger stone and soil, lies a basin formed around 2.1 billion years ago. The rocks of the Francevilian Basin were once mud and sediment on the floor of an ancient sea. They preserve evidence of oxygenated water, unusual carbon cycling, and structures that have forced scientists to ask whether complex life appeared far earlier than expected. The structures are difficult to dismiss at first glance. Some are broad and disc-shaped, others have lobes, folds, or elongated forms. A few reach several centimeters across, far larger than ordinary single cells. They occur in groups within particular sedimentary layers, as though something favored a specific environment. Researchers who support a biological origin argue that the structures contain organized internal textures and carbon-rich material consistent with life. Their distribution may reflect communities living on or near the seafloor. The surrounding sediments suggest calm, shallow marine conditions with enough oxygen to support more active metabolism. If this interpretation is correct, the Francevilian forms represent an astonishing experiment. They would predate the familiar ediacaran organisms by roughly 1.5 billion years. Complex or multicellular organization would not be a late event near the dawn of animals. It would have appeared during the aftermath of Earth's first oxygen revolution, but ancient rocks are skilled at imitation. Minerals grow into shapes that resemble cells. Sediments deform before hardening. Gas escapes through soft mud. Crystals replace organic material. Pressure and chemical fluids alter structures long after burial. A form that looks biological can emerge without any organism creating it. They point out that extraordinary claims require evidence able to survive every non-biological explanation. The debate remains open. This uncertainty is not a weakness in the story. It is the story. The structures occupy the boundary between what the rocks clearly preserve and what the human eye wants to recognize. They invite the same temptation that appears throughout ancient life research, to turn suggestive shapes into ancestors. A careful documentary must allow the wonder without forcing certainty. The Francevilian forms may record colonial organisms, coordinated microbial communities, or an early form of multicellularity. They may also include structures whose origins are partly or entirely geological. What is clearer is the environment around them. Geochemical evidence indicates that the basin experienced oxygenated conditions unusual for its time. Organic matter was abundant. Nutrient flows may have supported high productivity. The setting belonged to a temporary period when Earth's surface chemistry had expanded beyond the older anaerobic state. The basin could have been an oxygen oasis on a much larger scale than the microbial patches of the Archean. Within such a habitat, cells had room to experiment. Multicellularity does not begin with an animal body. It can begin when cells remain attached after division, coordinate their behavior, share nutrients, or specialize in simple ways. A colony may become large enough to move through gradients differently from isolated cells. Its outer cells meet the environment while inner cells experience protection. Larger size can provide benefits. A colony may resist being consumed, reach new food sources, or remain stable on sediment. It can also create problems. Oxygen and nutrients must move inward. Waste must move outward. Cells must cooperate rather than compete destructively with their neighbors. The Francevilian structures, if biological, may represent one solution attempted during a favorable interval. Their shapes suggest growth patterns more organized than random microbial films, yet nothing proves they were ancestors of later animals, plants, or fungi. They may belong to a branch that ended. This possibility changes the emotional shape of evolution. Complexity is often imagined as a single, slow ascent. Simple cells become complex cells, which become colonies, organisms, animals, and finally intelligence. The rocks may instead preserve repeated beginnings. Life explores a new level of organization, succeeds locally, then loses the environmental support needed to continue. A false dawn can still be a real dawn. It simply does not last. If oxygen levels later fell and productivity declined, the Francevillian ecosystem may have contracted or vanished. Organisms dependent on oxygenated water would lose habitat, nutrient limitation would reduce growth, anoxic or sulfidic conditions could return to the basin. A lineage capable of larger organization might disappear before leaving widespread descendants. No dramatic extinction layer would be required. The environment would narrow, generation by generation, until the experiment ended. The rocks preserve no clear continuation from the Francevillian forms to later eukaryotic fossils. For more than 1 billion years afterward, macroscopic life remains rare or absent from the record. This gap is one reason skeptics resist interpreting the structures as complex organisms. If they were biological, why did the innovation not spread? The answer may be that evolution can produce a possibility before the planet can sustain it. Modern life contains many examples of traits appearing more than once. Multicellularity evolved independently in animals, plants, fungi, algae, and several microbial groups. Cooperation among cells is not a single invention owned by one lineage. Given the right conditions, it may emerge repeatedly. The Francevillian basin could therefore record an early independent experiment, unrelated to later animals. Its importance would not depend on becoming our ancestor. It would show that complexity was biologically possible much earlier than its eventual global success. Or the rocks may be warning us how easily pattern becomes story. That tension should remain unresolved as the documentary leaves Gabon. The viewer is not asked to choose a final answer, but to recognize the stakes. If the structures are geological, they reveal the limits of fossil interpretation. If they are biological, they reveal a lost world of complex organization that rose and vanished in deep time. Either possibility leads to the same next question. Oxygen existed. Complex cells may have existed. Larger biological structures may even have appeared. Why then did Earth wait so long before filling its oceans with visible life? The answer lies not in one missing invention, but in the difference between opening a window and keeping it open. Oxygen had entered the atmosphere. Cells had learned to use it. Mitochondria may already have transformed the energy systems of early eukaryotes. In at least one ancient basin, large organized structures may have occupied the seafloor. From a distance, the next step seems inevitable. It was not. For more than 1 billion years after the first great oxygen rise, the planet remained dominated by microscopic life. Visible organisms did not spread through the oceans in any obvious way. Animals did not appear. Forests did not cover the continents. The revolution had opened a door, but most of the biosphere remained on the other side. The first limit was distribution. Surface waters exchanged gases with the air and received oxygen from photosynthesis. Deeper water could remain isolated. As organic matter sank and decomposed, microbes consumed the available oxygen. Where circulation was weak, the gas disappeared before reaching the seafloor. Below the oxygenated surface, vast regions were anoxic. Some contained dissolved iron. Others became rich in sulfide produced by microbes using sulfate. Sulfidic water was especially difficult for many oxygen-dependent organisms because it combined a lack of oxygen with a toxic compound capable of interfering with cellular machinery. The ocean was not one habitat. It was a layered collection of chemical worlds. Nutrients created another limit. Photosynthetic life needed phosphorus and nitrogen. Eukaryotic cells required metals and trace elements for enzymes. Yet oxygen altered where those materials traveled. Sulfidic conditions trapped certain metals. Continental weathering supplied nutrients unevenly. A productive coast might exist beside a nutrient-poor open ocean. Complexity requires more than survival. A large organism must move food and gases across greater distances than a single cell. Outer cells may receive oxygen while inner cells remain deprived. Waste accumulates. Neighboring cells must coordinate growth, prevent conflict, and reproduce as a collective rather than as independent competitors. Even a simple multicellular body creates political problems at the cellular scale. Cells must agree, through evolved rules, when to divide and when to stop. Some may need to sacrifice their own reproduction to support the whole. Damaged cells must be removed. Signals must travel between regions. Structural systems must hold the body together while allowing it to change shape. These features do not appear automatically when oxygen rises. They require genes, regulatory networks, and long periods of selection in environments where larger organization provides an advantage. Oxygen itself can create non-linear effects. More is not always better in a simple way. Experiments with multicellular yeast have shown that oxygen availability can influence body size differently under different conditions. Limited oxygen may favor larger groups in some settings by changing metabolism or creating internal gradients. High oxygen may remove that advantage. Biology responds to context, not slogans. The lesson is that oxygen is an enabling condition, not a command. It can support energy-rich metabolism, but it does not instruct cells to become animals. It can make sterols and complex membranes possible, but it does not guarantee nervous systems or movement. It can expand habitable space while leaving nutrients, climate, and ecology too unstable for large bodies to persist. Predation may also have mattered. When one organism begins consuming another, size, armor, movement, and cooperation become valuable. Ecological arms races can drive innovation. In a world dominated by microbial mats and dissolved nutrients, the advantages of large bodies may have been limited. Sexual reproduction, genome organization, developmental control, and communication among cells each added further steps. Some evolved early, others appeared repeatedly in different lineages. Many experiments probably vanished without fossils. Chance remained part of the process. A useful mutation must arise in a population capable of surviving long enough for it to spread. A promising lineage can disappear through climate change, nutrient collapse, or local catastrophe. Evolution does not preserve every successful idea. The Francevillian forms, if biological, may illustrate this fragility. They could have developed within an oxygenated, nutrient-rich basin during a temporary window. Their organization may have worked there under those particular conditions. When the environment changed, the lineage may have had nowhere else to go. Complexity appeared locally before it became globally sustainable. This distinction explains why the oxygen catastrophe could engineer the possibility of complex life without producing an immediate complex world. Engineering in this sense did not mean design. The catastrophe changed energy budgets, mineral cycles, climate feedbacks, and ecological boundaries. It created raw conditions. Evolution still had to assemble the machinery. The long delay also protects the story from a human-centered ending. Oxygen did not rise because animals were meant to arrive. Mitochondria did not form because intelligence waited in the future. Most evolutionary paths never led toward large bodies or consciousness. Microbial life remained successful and still dominates much of Earth's chemistry. Human beings are one late outcome among countless possibilities. During the long Middle Proterozoic, continents moved slowly through changing oceans. Eukaryotic lineages diversified. Algae altered food webs. Microbes continued regulating carbon, sulfur, nitrogen, iron, and methane. The planet prepared no stage, yet the accumulation of innovations gradually changed what could survive. Later oxygen rises would expand habitable depths. New nutrient conditions would increase productivity. Ecological interactions would become more intense. By the time animals appeared, they inherited a world built through billions of years of microbial labor and crisis. The false dawn in Gabon, whether biological or geological, leaves a useful mystery. It reminds the viewer that evidence of possibility is not evidence of inevitability. A planet can approach a threshold, cross it locally, then retreat. Complex life required the door to remain open long enough for many separate innovations to meet. The oxygen revolution had changed the rules. It had not written the final result. That result would depend on later worlds, later accidents, and lineages no cyanobacterium could have predicted. Yet the catastrophe was no longer distant history. Its chemical bargain survives inside every complex cell. To understand how completely the ancient crisis entered modern life, the story must return to the present and follow a single breath inward. A breath begins before it is noticed. Air enters through the nose or mouth, moves down the throat, and divides through smaller passages inside the lungs. At the ends of those passages, oxygen crosses a surface thin enough for molecules to pass. Blood carries the gas away, binding it to proteins and delivering it through the body. Nothing about this feels ancient. It happens in seconds. Yet every stage depends on a planetary crisis more than 2 billion years old. The oxygen entering the lungs was first released into Earth's surface environment, by organisms using sunlight to split water. The system inside plant chloroplasts and modern cyanobacteria descends from that discovery. Before leaves existed, microbial cells were already performing the reaction in shallow seas. Most of their oxygen vanished into iron, gases, organic matter, and rock. Later, the balance changed. The atmosphere gained a persistent supply. Anaerobic ecosystems contracted. Methane declined. Ice spread. Survivors adapted, and some learned to use the new gas. The body continues that history. Oxygen bound in the blood reaches tissues and moves into cells. There, mitochondria guide electrons through membrane-bound reactions. Oxygen accepts them at the end, helping form water while the cell captures energy. Muscles contract, nerves transmit signals, wounds repair, and organs maintain their internal order through energy linked to that final chemical acceptance. The mitochondria performing this work descend from bacteria that once lived independently. Every complex cell contains the altered remains of an ancient partnership. Human energy is therefore not produced by a purely human invention. It is produced through a merger older than animals, preserved inside each generation. The story becomes stranger when oxygen's danger is considered. The body needs the gas, but too much exposure damages tissue. Too little causes cells to fail. Reactive oxygen molecules form as normal byproducts of metabolism. Enzymes neutralize them. Damaged proteins are replaced. DNA is repaired. Antioxidants and cellular compartments limit uncontrolled reactions. Modern life has not conquered oxygen. It manages oxygen continuously. This management carries the memory of the catastrophe. Ancient cells faced a spreading poison and evolved defenses. Later organisms inherited and expanded those defenses because the gas became essential. The systems protecting a human cell from oxidative damage are distant answers to an environmental problem that began in microbial oceans. Even aging is partly connected to the tension. Metabolism produces energy, but it also creates molecular stress. Inflammation uses reactive chemistry to fight infection, yet uncontrolled inflammation harms the body. Cells deliberately produce oxygen-based molecules for signaling and defense, then work to prevent those molecules from attacking everything nearby. The old poison became a messenger, a weapon, and a fuel. This does not mean the Great Oxidation Event directly caused every modern trait. Billions of years separate the first atmospheric rise from human biology. Evolution added layers of innovation, loss, and chance. Later, oxygen increases were essential for widespread animal life. The early event established a durable foundation, not a finished design. Still, the foundation reaches everywhere. The energy used to understand this story depends on oxygen. The brain maintains electrical gradients through molecules produced by cellular respiration. Life accepted a dangerous reactant because the energy reward was immense. The bargain also shaped body size and activity. Large animals require systems that move oxygen rapidly from the environment to internal cells. Lungs, gills, blood vessels, respiratory pigments, and hearts solve the problem in different ways. They allow tissues far from the surface to participate in aerobic metabolism. No such systems were needed in the earliest microbial mats. A cell met its environment directly. As bodies grew, oxygen delivery became architecture. Evolution built tubes, pumps, branching surfaces, and molecules capable of binding the gas without allowing it to react freely. A human body is therefore an oxygen control system wrapped around trillions of cells. Yet beneath every oxygen-dependent tissue, anaerobic chemistry remains. The digestive system contains communities living where oxygen is low. The old and new worlds still meet inside living organisms. A breath also connects the present atmosphere to the biosphere producing it. Plants and algae release oxygen, but most atmospheric oxygen exists because organic carbon has been buried over geological time. If every product of photosynthesis immediately reacted again, no large reserve would remain. The breathable sky is a result of long-term imbalance stored in sediments, rocks, and fossil carbon. Burning ancient organic matter reverses part of that storage, consuming oxygen and releasing carbon dioxide. Production, burial, oxidation, and release remain linked. The atmosphere is not a permanent gift. It is an active biological and geological state. This realization changes the meaning of ordinary air. Oxygen is not simply present because Earth is naturally suited to animals. Earth became oxygenated through a sequence of biological disruptions. The gas exists because life altered the planet faster than rocks and other organisms could completely undo the change. The first beneficiaries were not humans. Many early organisms suffered. Some disappeared. Others retreated into places where oxygen could not reach. The modern breath carries the consequence of their lost habitats. There is no need to turn this history into guilt. Evolution contains no moral balance sheet. But the contrast remains powerful. The same atmospheric transformation that likely destroyed much of an ancient biosphere eventually became necessary for the consciousness capable of reconstructing it. Every breath is built from both extinction and survival. When oxygen enters the lungs, it crosses from the outer world into a lineage of cells that has spent billions of years learning how to contain it. The gas passes through blood, enters mitochondria, accepts electrons, and becomes water. The journey ends quietly. But beyond Earth, astronomers search for the same molecule in the atmospheres of distant planets. They hope oxygen may reveal life. Ancient Earth offers a warning. Oxygen can indicate biology, catastrophe, or a world still waiting for complexity. The next breath may therefore be more than a connection to the past. It may be a clue to how life announces itself across the universe. Section 2. The breath of another world across the darkness beyond Earth. Thousands of planets circle other stars. Most are too distant to see as worlds. Astronomers detect them through the slight dimming of starlight, the motion of a star pulled by an unseen companion, or the faint separation of planetary light from the brilliance around it. The next challenge is not merely finding planets, it is learning what fills their skies. When a planet passes in front of its star, part of the starlight may travel through the planet's atmosphere. Molecules absorb particular colors. With sensitive instruments, astronomers can search those patterns for water vapor, carbon dioxide, methane, ozone, and oxygen. Oxygen attracts attention because, on modern Earth, nearly all of it is connected to life. Plants, algae, and cyanobacteria continually produce the gas through photosynthesis. Without biological replacement, oxygen would react with rocks, gases, and organic matter and gradually decline. A distant atmosphere rich in oxygen might therefore seem like a clear announcement. This planet is alive. Ancient Earth warns that the message would be more complicated. For much of Earth's inhabited history, the atmosphere contained almost no oxygen. An astronomer observing the planet during the Archean might detect methane, carbon dioxide, haze, and water, but miss the thriving microbial biosphere below. Life could be abundant while remaining almost invisible to an oxygen search. Later, during the Great Oxidation Event, oxygen would begin appearing while the planet remained dominated by microbes. A distant observer might detect the gas and imagine forests, animals, or complex ecosystems that did not exist. Oxygen could reveal photosynthesis without revealing complexity. There is another problem. Oxygen can form without life. Ultraviolet light can split water vapor in an atmosphere. Hydrogen, being light, can escape into space, leaving oxygen behind. Carbon dioxide can also be broken apart by light, creating oxygen under some circumstances. A lifeless planet might therefore display the gas most closely associated with living Earth. Astronomers must search for context. Is liquid water possible at the surface? What kind of star does the planet orbit? Does the atmosphere contain carbon monoxide, methane, or other gases that change the interpretation? One powerful clue is disequilibrium. An atmosphere is in chemical disequilibrium when it contains substances that should react with each other and disappear, yet remain present because something continually replaces them. Modern Earth holds abundant oxygen alongside methane. The gases react, so their coexistence indicates active sources. Life is not the only possible source, but the combination can be harder to explain without biology than either gas alone. Again, ancient Earth complicates the picture. Before oxygen rose, methane and carbon dioxide may have carried the stronger biological signal. During transitional periods, local oxygen production could remain hidden from the atmosphere. A successful search for alien life must therefore recognize worlds at different stages, rather than looking only for copies of present Earth. The Oxygen Catastrophe becomes a lesson in planetary timing. A planet may be alive before it becomes breathable. It may become oxygenated before complex organisms appear. It may suffer climate collapse because of the gas. It may overshoot, lose oxygen, and remain limited for 1 billion years. The presence of life does not guarantee stability, and the presence of oxygen does not guarantee intelligence. This changes the emotional meaning of distant detection. Finding oxygen around another world would not be the end of the mystery, it would be the beginning. Astronomers would ask what produces it, what consumes it, how long it has been present, and whether oceans or continents support continuous cycling. Perhaps the planet would resemble Earth before animals, covered in microbial seas beneath an unfamiliar sky. Perhaps oxygen-producing organisms would be entering their own catastrophe, pushing anaerobic life into hidden refuges. No telescope currently reveals such detail across interstellar distance, but the questions are already shaped by Earth's past. The planet beneath human feet is the only inhabited world known, and its history shows that habitability is not a fixed condition. It is a moving relationship among life, rock, ocean, air, and star. The first mass extinction engineered by oxygen, if that phrase is used carefully, was not a single massacre preserved in fossils. It was a long contraction of environments suited to oxygen-sensitive life. Shallow seas changed, chemical refuges narrowed, methane weakened, climate cooled. Survivors retreated, adapted, or learned to use the poison. From that destruction came opportunity, but not by design. Oxygen created new energy pathways. It changed minerals and nutrient cycles. It helped make sterols and complex membranes possible. It supported aerobic bacteria and later mitochondria. It expanded the energy available to eukaryotic cells. Yet it also produced reactive damage, glaciation, ecological collapse, and long periods of limitation. Creation and catastrophe were not opposite stories. They were different views of the same process. A cyanobacterial cell releasing oxygen more than 2 billion years ago could not know what followed. Its waste entered iron-rich water and vanished. Another cell repeated the reaction, then another. Across uncounted generations, the gas altered oceans, rocks, climate, and life. Most of the organisms affected left no recognizable fossil. Their world survives only through chemical traces and descendants living in anoxic places. The silence of the record is part of the mystery. Human beings arrived unimaginably later. They learned to read sulfur isotopes, date ancient minerals, compare genes, and search the atmospheres of planets they cannot visit. Their bodies depend on the same oxygen whose arrival likely devastated the first biosphere. The circle closes with another quiet breath. Air enters, oxygen reaches cells, mitochondria use it. Water forms. The body continues for another moment because ancient life changed the sky. Beyond that sky, other worlds turn through darkness. Some may be lifeless. Some may hide life beneath atmospheres no one has learned to recognize. A few may be approaching their own chemical threshold. The greatest beginnings may not arrive as gentle progress. They may appear as pollution, imbalance, extinction, or cold. Only much later does the new world reveal what the catastrophe made possible.